The surface design project aimed to determine how changes in basement membrane affect the deep structure of tumors. When cultivated on a non-compliant, two-dimensional, chemically inert surface, mammary epithelial cells that become transformed in breast cancer fail to achieve a normal form, even though they possess the appropriate genes that should enable them to do this. Essentially, they form flat monolayers that resemble many other cell types. When cultivated within a compliant 3D ECM within a tissue culture dish, however, cells can be induced to undergo a normal morphogenetic process that resembles the breast in vivo. They form a basement membrane, a lumen, the cells become polarized. Once they have achieved this state, the breast epithelium will remain quiescent. With cancer, however, the integrity and quality of the external surface changes, resulting in an inappropriate growth response to this modified surface matrix environment. Understanding precisely how this occurs is a critical step in comprehending this disease, since it is now appreciated that changes in the normal surface architecture of breast epithelial tissue at the level of the extracellular matrix can directly initiate and propagate tumor formation. This is a profound discovery, since it suggests that by reversing cell ECM interactions within tumors, cancer cells can be tricked into normal modes of behavior.
As mentioned, for the surface design project, we sought to quantify and spatialize cellular and tissue contour information using normal human mammary epithelial cells cultivated within a 3D normal or tumor-like microenvironment. Specifically, the project examined morphogenesis, lumen formation, and nuclear packing behavior as a response to alterations in the tissue surface design. These parameters were chosen since they represent cardinal features of aggressive forms of this disease. Additionally, these relationships were tested against and funneled through simple architectural and deployable structures. Here, the algorithmic and digital exploration of relationships between interacting cells and their immediate tissue environment gives rise to an abstract yet deeper understanding of architectural form as it relates to a dynamic boundary condition and existing constraints found within the design mechanisms of deployable structures. In turn, this information has provided new clues as to how the tissue microenvironment may impact cell behavior in breast cancer.
For the second project, we investigated the role of the microenvironment in controlling smooth muscle cell motility and the shifting geometries inherent in this movement. In brief, with pulmonary vascular disease, the ECM microenvironment within the blood vessel wall changes radically. Collagen fibers are broken down, causing cells to move from their resting place into the lumen of the blood vessel, resulting in occlusion, pulmonary vascular disease, and ultimately heart failure. So how do these cells move in response to changes in their extracellular matrix, and can knowledge of this altered movement be used clinically? We reasoned that by examining and abstracting the cellular edge behaviors of cells within a normal or diseased tissue microenvironment, we'd be able to generate novel ways of quantifying, visualizing, and characterizing cell motility on a patient-to-patient basis. Through computation and abstraction of edge behaviors, we aim to derive personalized signatures for patients with pulmonary vascular disease.
On the architectural and biomedical levels, this project has resulted in a more complex and quantitative understanding of a dynamic cellular structure that negotiates and integrates changes in external forces with internal cellular mechanics. For the final project, we investigated part-to-whole relationships during capillary formation by lung endothelial cells. At the biological level, coordinated cellular networking is required to form the exquisite fractal network that emerges in the developing and mature lung to facilitate efficient gas exchange from birth onwards. Real-time imaging of endothelial cells cultured within specialized ECM microenvironments that either suppress or promote networking formed the basis of this project. At the architectural design levels, the examination of this dynamic cellular networking system through diagramming and abstraction has provided a new approach to the examination of programming, form, structure, and experience. Additionally, we are able to examine how part-to-whole relationships are articulated through matrix-driven acts of nonlinear fabrication.
What is now clear to us from these initial investigations is that there is room to develop models which capture and cultivate the dynamics of the less obvious systems of architecture and the more obvious living complexities of biological systems. Like mammary epithelial cells cultivated in a 2D environment which produces behavior and information that is insufficient when compared to the actual in vivo 3D system, perhaps architecture can take a cue from biology in matching the complexity of its generative design models to the very dynamic features of the living environment and organic milieu in which the architecture is a part.
By pairing architects with cell biologists in complex biological design problems and abstracting the inherent relationships into code-driven, parametric, and associative models, it is possible to gain new insights into how nature deals with nonlinear behavior within cell and tissue structures. Perhaps architects might learn from these biological models, such that architecture acquires tissueness or cellness and is not merely cell or tissue-like. It is through these interdisciplinary pursuits that we are exposed to new modes of thinking, thus affording new models for architecture and biomedicine, and a realization that we are all searching for a deeper understanding of form and function, be it real or abstract.